Hello everyone, my name is Edo Danielian, one of the students in Biology Department in the Faculty of Science, Chulalongkorn University. This seminar is under supervision of Assistant Professor Dr. Chachawan Chaisukul. In this opportunity, I would like to present the evaluation of Cyphocleonus trisulcatus, which belong to Curculionidae family, as a potential biological control agent for Leucanthemum vulgare in North America. This seminar based on research by Stutz et al. In 2019. Leucanthemum vulgare, or oxeye daisy, is perennial forb native to Europe and Western Asia. Oxeye daisy has been introduced to many parts of the world as an ornamental plant. Now, it can be found on all continents except Antarctica. Oxeye daisy is invasive in several parts of the world, especially in North America. It is become invasive because the absence of natural predators in new habitat. In nature, oxeye daisy has several impacts. It is able to form dense, extensive population in pastures, where it is generally avoided by grazing cattle. Oxeye daisy has shallow root system, and this type of root can promote soil erosion. Not only that, it is also reduce plant diversity. In this study, main non-target species is Shasta daisies, and the native plant in North America, which is Matricaria occidentalis. And there are three varieties of Shasta daisy as a non-target species, where the first one is Leucanthemum superbum as Alaska, and then the second one is crazy daisy, and the third one is Leucanthemum ex maximum. There are some methods to control oxeye daisy. The first one is manual control. It is usually referred to pulling or digging. The second one is mechanical control. It is usually referred to the method where require mower. And the third one is chemical control, where it is obviously using chemical compound such as herbicides. The previous method have some weaknesses. Manual and mechanical control require high resources, such as people and tools, and it is not suitable in large scale and in the remote area. Not only that, manual and mechanical control is possible to spread the seed to another places. And we have chemical method for controlling the invasive plant. However, it is limited use in environmental sensitive area. There is one more method for controlling invasive plant species, that is biological control. Biological control agent is a natural enemies for controlling plant, like parasite, predator, or other mechanism. One of the potential insects as a biological control agent for oxeye daisy is Cyphocleonus trisulcatus. Cyphocleonus trisulcatus is root-feeding weevil. Should we introduce C. trisulcatus to North America as biological control agent? Before biological control agent is being released to nature, it is important to evaluate the impact and characterize the natural host. We have to make sure that it's not dangerous to agricultural plant. The main non-target species of concern is the horticultural Shasta daisy. And the objective of this study is to evaluate this potential biological control of oxeye daisy. In this study, there are 4 methods to evaluate potential biological control agent. First is impact experiment. Second is no choice oviposition and larval development test. Third is multiple choice oviposition and larval development test in a field cage. The last is multiple choice oviposition and larval development test in open field. In the impact experiment, 30 pots of oxeye daisy were used. Oxeye daisy were separated into two groups. First group, as a control, were kept free of attack by this weevil or Cyphocleonus. And the second group was attacked by Cyphocleonus. The objective of the experiment is to evaluate the impact of Cyphocleonus to oxeye daisy. For the statistical analysis in impact experiment, they use quasi-Poisson generalized linear model for number of shoot and number of flower head, and linear model for all other parameters. In the second method for evaluating potential biological control agent, where as no choice oviposition and larval development test, in this experiment, 50 different plant species were used. Each species were placed into the pot, and after that, 2 egg-laying female of Cyphocleonus were placed onto individual potted. After 3 to 8 weeks, plant had been exposed with Cyphocleonus. Roots were dissected and checked for larvae. They used this method to know the probable host for Cyphocleonus. The next is multiple choice oviposition and larval development test in a field cage. 4 species were used in this experiment. The species were chosen based on the second experiment results, and some of them are native to North America, which is Matricaria occidentalis and commercially grown Matricaria chamomilla, and Coleoptera coronaria. White circle in here is a representative of Leucanthemum vulgare or oxeye daisy. Yellow circle is a representative of Matricaria chamomilla, and a black circle is a representative of Matricaria occidentalis, and a blue circle is a representative of Coleoptera coronaria. There are 4 large ghosts covered field cage with dimension 4 meter square area and 1.6 meter height. In this experiment, Leucanthemum vulgare or oxeye daisy were used as a control. After 10 days of female Cyphocleonus released, all plants were removed from the cage and checked for females. Once all females were retrieved, all plants individually placed on pot and covered with ghosts. After 1 month, then roots were dissected and the total number of larvae were recorded. In multiple choice oviposition and larval development tests in open field, these tests were set up with 4 different Shasta daisy varieties. To investigate whether Cyphocleonus were actively searched for Shasta daisy, the experimental patches were a bit like this. 12 potted plants of each of the 4 Shasta daisy varieties and 48 plants of Leucanthemum vulgare or oxeye daisy, where 66 plants in the rosette stage and 12 others with shoots. Each patch consisted of first Shasta daisy. The black dot in the middle is a release point of Cyphocleonus, and the patches were set up at 2.5, 5, and 10 meters radially from release point. The potted plant were buried into the soil so that the upper edge of the pot was level with the surface of the soil. 1 month later, all plants were checked for Cyphocleonus and all plants regularly checked for adult emergence. The first results in this study is impact experiment. The chart A is above ground biomass. We can see the different significance of controlled biomass and infested plant biomass. By infesting the C. trisulcatus, the decrease of biomass could reach around 44%. In chart B, we can also see different below ground biomass of control plant and infested plant, and the reducing of below ground biomass could reach around 68%. For the chart C, it is the comparison of number of flower head between control and infested plant. We can see the significant difference also in here. But in chart D, it is the comparison of length of the longest shoot between control and infested plant. However, the result showed that there is insignificant difference. The second result is from no-choice oviposition and larva development test. 3 weeks after plant have been exposed to C. trisulcatus, the root of plant were dissected. Larvae found in 5 plant species, which are first one is Leucanthemum vulgare or oxeye, the second one is Glebionis coronaria, and the third one is Matricaria chamomilla, and the fourth is Matricaria occidentalis, and the fifth is Achillea tarmica. In the third results from the multiple choice oviposition and larva development test in a field cage show that the C. trisulcatus had tendency to produce egg on Leucanthemum vulgare or oxeye with rosettes instead of in Leucanthemum vulgare without rosettes. And C. trisulcatus or this weevil also lay their egg in native plant species. The last results from multiple choice oviposition and larval development tests in open field cage show that Leucanthemum vulgare or oxeye without rosette were less likely being attacked by C. trisulcatus. We can see in here in the yellow highlight. And then compared to the Leucanthemum vulgare with rosette in red oval here, where more likely being attacked by C. trisulcatus. In green highlight, all of the tested Shasta daisies heavily attack up to 10 meters from release point. It indicates that C. trisulcatus search for Shasta daisy under natural condition in North America. From the results, we can see that C. trisulcatus severely damaged oxeye daisy. As we know that C. trisulcatus more likely attack oxeye daisy with rosettes, it is indicate that C. trisulcatus could suppress invasive oxeye daisy by reducing sexual reproduction. And the similar impact of C. trisulcatus should be expected if it were released as biological control agent. C. trisulcatus is able to develop on Shasta daisy. From the result, C. trisulcatus would likely attack Shasta daisy in North America. And the damage on Shasta daisy is comparable to damage on oxeye daisy. The implication for the biological control of oxeye daisy. C. trisulcatus is not suitable biological control agent in North America. However, C. trisulcatus would be suitable in the continent where Shasta daisy is not as popular as in North America. We can conclude that C. trisulcatus severely damaged oxeye daisy. It can be used as biological control agent of oxeye daisy. However, it's not suitable to be biological control agent in North America. I would like to express my special thanks of gratitude to Assistant Professor Dr. Chachawan Chaisakul for his able guidance and support in planning and completing of my preparation for the seminar. Thank you.